Carbohydrate metabolism, gluconeogenesis. In this slide presentation, we will review gluconeogenesis, the process of forming glucose from non-carbohydrate sources such as lactate, pyruvate, oxaloacetate, glycerol, and glucogenic amino acids. It is important to note that gluconeogenesis occurs only in the liver. The steps involved in gluconeogenesis are essentially the reverse of glycolysis, with glycolysis being the conversion of glucose to pyruvate. However, with gluconeogenesis, we have the conversion of pyruvate back to glucose. It is important to note that acetyl-CoA cannot be converted to glucose in the process of gluconeogenesis. This is because the pyruvate dehydrogenase complex is not reversible, meaning that we can convert pyruvate to acetyl-CoA, but we cannot convert acetyl-CoA to pyruvate. Like with glycogenesis and glycogenolysis, it will be important to discuss the metabolic environment promoting gluconeogenesis. Similar to glycogenolysis, gluconeogenesis allows for mobilization of glucose to the blood when blood glucose drops in a fasted state. Additionally, gluconeogenesis promotes glucose mobilization and increased ATP production during exercise. Therefore, gluconeogenesis will be more likely to occur during a fasted state or during exercise. The regulation of gluconeogenesis occurs at the steps of the irreversible reactions of glycolysis. Just as a reminder, we can review here the irreversible steps or irreversible reactions of glycolysis. So first we have step 1, which is glucose to glucose 6-phosphate, and you can see the one directional arrow. Step 3, which is fructose 6-phosphate to fructose 1,6-bisphosphate. And step 9, which is phosphoenolpyruvate to pyruvate. Also here, you can review the irreversible reactions of glycolysis, the reactants, the products, and their enzymes. For gluconeogenesis to occur, each of the irreversible reactions will need to be bypassed. Therefore, we'll need to bypass pyruvate kinase, PFK, and glucokinase. The enzymes allowing for each of the irreversible steps to be bypassed are active specifically in the liver. We will go over each of these enzymes and the process involved in bypassing each of the irreversible steps. Bypassing PFK and glucokinase involve the activity of phosphatase enzymes. While kinases active in glycolysis add a phosphate group, for example, we see glucokinase adds a phosphate group to glucose, converting it to glucose 6-phosphate, whereas PFK adds a phosphate group to fructose 6-phosphate, forming fructose 1,6-bisphosphate. So in contrast to the kinases, the phosphatase enzymes remove a phosphate group. The activity of these phosphatase enzymes are illustrated in this diagram. So you can see in this diagram, the arrows pointing down are those of glycolysis, and the arrows pointing up are those of gluconeogenesis. So you can see through glycolysis, the kinase enzymes add a phosphate group from ATP. Here we see conversion of glucose to glucose 6-phosphate with glucokinase. And here we see conversion of fructose 6-phosphate to fructose 1,6-phosphate bisphosphate with phosphofructokinase. And again, the phosphate group is coming from the ATP. In contrast, you can see the activity of the phosphatase enzymes. So this specific phosphatase enzyme, active in gluconeogenesis, is fructose 1,6 bisphosphatase, which removes a phosphate group from fructose 1,6 bisphosphate. We see the removal of the phosphate group, converting it to fructose 6-phosphate. The second specific phosphatase enzyme is glucose 6-phosphatase. Removing a phosphate group from glucose 6-phosphate, you see the phosphate group being removed, converting it to glucose. Therefore, you can see how these specific phosphatase enzymes aid in bypassing steps 1 and steps 3 of glycolysis during gluconeogenesis. The process of bypassing pyruvate kinase, which is active in step 9 of glycolysis, is more complex and involves more than a phosphatase enzyme. Pyruvate carboxylase enzyme is the primary enzyme involved in the process of bypassing pyruvate kinase. However, there are a series of steps occurring in both the cytosol and the mitochondria of the cell, which these steps are involved in the bypassing of pyruvate kinase. You can see the steps in this diagram. Here we see phosphoenolpyruvate to pyruvate, catalyzed by pyruvate kinase, and here are the series of steps involved in bypassing pyruvate kinase. We see involvement in the cytosol and also in the mitochondria. First, we have pyruvate moving into the mitochondria, and then we have conversion of pyruvate to oxaloacetate, oxaloacetate to malate, malate leaving the mitochondria, and then being converted back to oxaloacetate, and the oxaloacetate is then converted to phosphoenolpyruvate. Here we then see the additional steps of gluconeogenesis, the activity of the phosphatase enzymes, and the eventual conversion to glucose. We will now discuss a specific type of gluconeogenesis, a cycle called the Cori cycle, which involves interaction between compounds produced in the muscle and the liver. During anaerobic exercise, for example, pyruvate is converted to lactate. This lactate can be recycled to form glucose in the liver, which can then be used again to fuel the muscle. Here we see in this diagram that the lactate formed in the muscle during anaerobic exercise will exit the muscle cell, enter circulation, and will then be taken up by the liver. Those 2 lactate then will be converted to 2-pyruvate, and then in the process of gluconeogenesis, the 2-pyruvate will be converted to glucose. The glucose formed in the liver through gluconeogenesis can then exit the liver cell, enter circulation, and then be picked up by the muscle again. And that glucose can be used to undergo glycolysis, produce ATP to fuel continued muscle contraction. This is an energetically expensive process, as you can see the conversion of lactate to pyruvate to glucose utilizes 6 ATP. This diagram also portrays the Cori cycle with the conversion of glucose to lactate in the muscle, and then lactate to glucose in the liver, and then the glucose then being utilized again in the muscle. However, it further shows that the glucose in the muscle can be obtained by the breakdown of muscle glycogen during the process of glycogenolysis.